And I'm pleased to introduce our first presentation for this morning. Dr. Eric Rellinger is an assistant professor at Division of Pediatric Surgery at the University of Kentucky. His presentation is entitled GDP mannose 4,6 dehydratase is a key driver in MYC, amplified neuroblastoma, core fusilation, and tumorigenesis. That was hard.
It was perfect. Good morning, everyone. Thanks for the opportunity to share our work. As we all know, neuroblastoma is one of the most common extracranial solid tumors in children. It's a histologically heterogeneous disease and also clinically heterogeneous. And those children with high-risk disease continue to have dismal survival despite induction chemotherapy, surgery, stem cell transplantation, radiation therapy, and immunologic therapy, most commonly with anti-GD2-based therapeutics. About 40% of those children with high-risk disease feature MYC amplification. When I worked in the laboratory of Dr. Dae Chung, I worked with a couple of MYC mavens who revealed to me the biologic significance of NMYC. It's an oncogenic transcription factor that turns on a myriad of genes. And the other thing that it does is it metabolically transforms cancer cells, alters the way in which they use sugars, amino acids, and fatty acids. And when I transitioned my career to the University of Kentucky, I joined the Center for Cancer Metabolism and started working with two glycobiologists. These are folks who study complex carbohydrates and their role in biology. This is Dr. Ramon Sun and Matt Gentry. And what they revealed to me, something that changed my view on biology, is that sugars are important. So all your cells are covered with sugars, and in particular, what I'm going to talk about are glycans, which are the sugars that are attached to proteins and lipids, and they have biologic significance. The way in which they shape my view of biology is by expanding my perception of the central dogma. So when I went through medical school, it was DNA to RNA to protein, and then there was some stuff about epigenetic regulation upstream, but there's been far less discussed about what happens downstream, post-translational modifications, altering the way that proteins are localized, how they're activated, how they're degraded. And there's 10 different million sugars, and the pattern in which they're added on to proteins, we really don't actually understand what the significance of these sugars are. So when you think about sugars adding complexity to the existing central dogma, it adds enormous complexity in both organisms and also disease progression. As I delved into this a little bit more, in adult cancers, it's well known that glycosylation is different. In particular, there's some people consider glycosylation to be a hallmark of cancer, and high mannose, sialic acid-rich, and core fucosylated glycans are actually common features of many adult solid tumors. So we created the hypothesis that if we could define the end-link glycome of MYC-amplified neuroblastomas, we could actually identify novel and actionable metabolic vulnerabilities to allow us to design treatments for children. The big driver behind this is the fact that I was introduced to new technology, MALDI-MSI, which allowed me to profile the sugars in human tissues, which was a new insight into biology that previously I don't think we've had access to. So in MALDI-MSI, you can take a formalin-fixed paraffin-embedded sample, take one, send it to the pathologist, and have it histologically annotated for different regions. And then the other one gets sent to a robotic sprayer where enzymes added that cleaves the glycans, and then a matrix that keeps it preserved in its normal heterogeneity. And then it passes through the mass spectrometry imager, and it allows you to create a heat map for these different glycans throughout the tumor tissue. This is an example of the MALDI-MSI, and it's kind of like a typewriter. It moves along the tissue sample, and each place where there's a peak represents a glycan. So it allows you to create a heat map and preserve the heterogeneity of your tumor samples throughout the tumor. So we then looked at the neuroblast-rich regions of MYC-amplified neuroblastomas, and we compared them to non-amplifieds. And what we found is that there were distinct sugar patterns in the MYC-amplified neuroblastomas. And those ones that reached significant, many of them actually had a consistent feature. So in this table here, these are all the significant glycans present in MYC-amplified neuroblastomas, and all of them have that little red triangle, or 6 of the 7 have that little red triangle, which is actually fucose, at the alpha-1,6 position, or fucosylation. So what the fucose? Like, why is a surgeon studying this? And it's interesting because fucose is actually central to your biology. Fucose is present in seaweed, and then your cells can either make it from mannose or glucose precursors through GMDS or GDP mannose 4,6 dehydratase and FX, or you can salvage it from existing glycoconjugates or take it in through the serum. We started looking at parts of the fucosylation pathway, and when we looked at GMDS, or that mediator of de novo synthesis, what we found was that high levels of GMDS were associated with poor neuroblastoma survival in public available data sets, advanced stage disease, and also MYC amplified neuroblastomas. So given the role that MYC plays in turning on genes as an oncogenic transcription factor, we started to dissect this, and we used tetracycline-inducible systems to turn MYC on and turn it off. And then also, and we found that when you turn the MYC on, GMDS goes up, and when you turn MYC off, GMDS goes down. And we also found that MYC binds directly to the GMDS promoter through promoter luciferase assays and chromatin immunoprecipitation. We then wanted to dissect how this de novo enzyme, synthesis enzyme, altered core fucosylated abundance. So we used lectins, which are proteins that bind to sugars, to show that when we blocked GMDS, the core fucosylated glycan abundance decreased on the whole cell, on the cell surface, and what's actually excreted from the cell itself. We then wanted to determine what's the significance of this enzyme in mediating neuroblastoma tumorigenesis. So we took those knockdown cells, looked at them in a dish, and they don't adhere as well, they don't migrate as well. And then when you inject them into a mouse, they form fewer and smaller tumors. So this led us to hypothesize, well, what happens when you treat a neuroblastoma and block fucosylation in an established tumor? And there's actually chemical compounds out there, there's about 6 or 7, that actually block fucosylation. So 2-fluorofucose is an orally bioavailable compound that actually will block de novo fucosynthesis and also fucosyl transferases. So we started supplementing 2-fluorofucose into the water of mice. First we checked it in the dish, sorry, I got ahead of myself, and we showed that 2-fluorofucose blocks fucosylation. And then we ended up treating the mice in situ. So we supplemented 2-fluorofucose into the water, and we found that neuroblastoma progression was impeded. The survival was increased when you treat these tumors to humane endpoints. And then, interestingly, if you look at panel C, you actually see large islands of necrosis that are induced within the tumors themselves when you actually defucosylate the tumors. So in summary, NMYC drives GMDS expression, leading to increased abundance of core fucosylated glycans in neuroblastoma. And our laboratory is devoted to determining the significance of these core fucosylated glycans in neuroblastoma biology, in particular focusing on how it may be regulating the tumor microenvironment to be used for novel therapeutic treatments. This work takes a team. I'm fortunate to work with folks from the University of Kentucky, Cincinnati Children's, and also the University of Florida. And I'm eternally grateful for the APSA Foundation for the opportunity to talk and the ability to support this work at a critical stage in my career. Thank you for the time and attention. I'd be happy to answer any questions that you may have.
I'd like to invite Dr. Joseph Church to come up to provide his presentation on artificial placenta support of newborn lambs with congenital diaphragmatic hernia. And he is an assistant professor of surgery at the University of Michigan. But one thing I would like to add about Dr. Rellinger's study is, I don't know that I'm intelligent enough to ask a question anyways, but it's a very nice example of how a team approach. So I'm not sure if you knew what fucosylation was until you met the marquee team at the Cancer Center there, but it's a great example of how, if you're susceptible to advice and opportunity, you can come up with a great project that you might not have thought about before.
Good morning, everyone. Thank you for that introduction, and thank you for the opportunity to present our work today. I have no financial disclosures except to express my gratitude to the APSA Foundation for the Grosfeld Scholar Grant in support of this research, so thank you. In congenital diaphragmatic hernia, the mainstay of support is gentle mechanical ventilation, but as many of us know, in 30% of neonates, this is insufficient, and they move on to cannulation to extracorporeal support or ECMO. During ECMO support, lung management generally consists of what we consider lung rest, trying to avoid atelectasis, but also avoiding volutrauma and barotrauma, allowing the lungs to recruit and grow and pulmonary hypertension to subside until eventually the lungs can participate once again in gas exchange and the child can be decannulated. However, this population continues to be characterized by 50% mortality, a number that really hasn't changed much over the last few decades. Causes of death include pulmonary hypoplasia and pulmonary hypertension that are refractory to this support and ECMO complications or some combination thereof. And so you have to ask, could there be a better method of ECLS to promote lung growth, alleviate pulmonary hypertension, and avoid complications? We hypothesized that maybe one type of ECMO approach would be an artificial placenta. Now, this consists of veno-venous ECMO with jugular venous drainage and umbilical vein reinfusion, avoidance of mechanical ventilation, preservation of fluid-filled lungs, in this case with perfluorocarbons, and preservation of fetal circulation. And our group, led by George Mialishka, has previously demonstrated in a preterm lamb model increases in alveolar development and type 2 pneumocyte density, surfactant production, and a decrease in lung injury during this support. So we had two aims related to this project as we apply it to CDH. One is to compare the artificial placenta to standard VA ECMO with rest settings in lambs with CDH in terms of lung growth and development, and then to go on and transition these lambs after this support to mechanical ventilation to better define pulmonary function and gas exchange. And this is a long and ongoing project. For the purposes of our discussion today, we'll focus on aim one. So first of all, to describe our methods, and I want to highlight the work here of Sarah King that you see in the upper corner, our research fellow and future pediatric surgeon who's really driven this work forward. We utilize the previously described surgical lamb model of CDH with a diaphragm defect created between gestational days 65 and 75. A large defect is created, stomach is pulled into the fetal chest, all incisions are closed, and pregnancy is maintained. We then deliver these lambs between gestational days 130 and 140, pretty close to term. And at delivery, they're either sacrificed for tissue control, either disease control, namely CDH, or an unoperated twins healthy controls, or cannulated to ECLS, either for the artificial placenta or VA ECMO. And we do perform these cannulations as exit procedures at this time. Just a little more detail on the comparison between the artificial placenta and VA ECMO. As mentioned, with the artificial placenta or AP, veno-venous ECMO, perfluorocarbons used as our liquid medium in the lungs. We also use a prostaglandin infusion to maintain ductal patency. Whereas in the VA ECMO group, we use the standard peripheral IJ carotid configuration. We use the 10-10-10 rest settings that many of us know, 10 of PEEP, 10 of rate, 10 of delta, and then do not use a prostaglandin infusion. But in both groups, use similar cannulas and circuit, umbilical artery and vein catheters, heparin for anticoagulation, and TPN for nutrition. Here's an example lamb on artificial placenta support. Once again, in both groups, lambs also receive as-needed sedation. We check regular blood gases, daily labs, and echocardiograms on days 1, 3, and 7 to assess ductal patency and cardiac function. This work is ongoing, but wanted to present kind of our work so far. And so you see here we have 7 CDH controls and 8 healthy controls. The inset numbers in the parentheses 3 and 6 are the data that you'll see presented today. For the others, we're looking into other methods of lung morphometric analysis and have kind of held that tissue aside. And then we've performed 3 runs with the artificial placenta and 2 with VA ECMO. As far as establishing the animal model, as you see here, this is an example of a CDH control at the time of necropsy with a substantial stomach and intestine in the left chest. You really don't even see the lungs as they're retracted over. And then on the right, with more of an abdominal view, you see a large diaphragmatic defect. This would be a study group type C. Just to show that there really is an impact in terms of hypoplasia from this model, as if anyone doubted it. I do love this picture. These are lungs from twins, one of which had a CDH created and one of which did not. I think you can probably guess which is which. This bore out in our lung weight to body weight ratios at the time of necropsy with significantly smaller lungs in the CDH group as compared to our healthy counterparts. Here's a summary of the data collected so far from our 5 extracorporeal runs, again 3 with the artificial placenta and 2 with VA. Unfortunately, with the artificial placenta, we have yet to make it the proposed 7 days. with termination of the experiment are between 4 and 6 days in all 3 runs. We'll get into some of the details of that just in a moment. Unfortunately, our second run was complicated by renal failure, which made the lamb very hard, difficult, and complex to support. The other 2 we suspect was related to right ventricular failure, although this is going to need some more investigation. Ductal patency was maintained in all 3 preps. And lung weight to body weight ratio actually did appear to resemble more closely that of the healthy controls at the end of the experiment as opposed to the CDH controls. Our first VA run, surprisingly, was cut very short by a tension pneumothorax despite using only rest settings and not any recruitment maneuvers. And our second run did make it the proposed 7 days with closure of the ductus by the end of the experiment and a lower lung weight to body weight ratio at that time. To summarize some hemodynamic and blood gas data over the course of the run with AP runs in green and VA ECMO runs in blue. Heart rate, pretty similar in the lambs between the 2 groups. You see a higher mean arterial pressure with VA ECMO compared to the artificial placenta, which makes sense given the systemic arterial inflow. Circuit flow, pretty similar between approaches, as was oxygen saturation, which hovered around 80% for the duration of support. I do want to highlight lactate, as I think this may be the clue as to what's going on with our artificial placenta-supported lambs. So if we focus in on the green trends, you see low and stable lactate until between days 4 and 6, when you have a fairly abrupt rise. And again, our hypothesis so far is this may be related to right ventricular strain on account of severe pulmonary hypertension and a slowly closing ductus, perhaps despite prostaglandin infusion. Of course, we're looking into this more thoroughly. So this is an ongoing and complicated set of experiments, and we're learning a lot along the way. Some of the lessons learned so far, I think, are first of all that premature lungs are not the same as CDH lungs. Both are characterized by some degree of immaturity, but the pathophysiology is clearly different, and therefore our approach to support may have some overlap, but is going to have some unique features. I think the artificial placenta is feasible for CDH, but cardiac strain is a concern, and we're going to need to solve this problem if we're going to keep pursuing this. I also think it was enlightening that rest ventilator settings may not be real rest. I know it's just one animal, but the fact that on rest settings without recruitment, you develop the tension pneumothorax is a bit telling, and it does make you scratch your head a little bit, perhaps, about what this may do in human lungs over the course of an ECMO run. And finally, whether you're looking at the artificial placenta or standard VA ECMO for CDH, I think we all know the devil's in the details. There are a lot of small things to adjust, a lot of tweaks, and which is why we're continuing to push this experimentation forward, because I don't think we can be disheartened over the fact that we've had these rises in lactate and right ventricular strain. It's going to take some turning of the knobs. Our next steps are to continue to push for 7 days of support. We're setting up for an extensive lung morphometric analysis comparing these 2 groups and our controls. Continue to aim to transition to mechanical ventilation to better evaluate gas exchange and lung compliance, and may introduce a set of lambs with a pulmonary vasodilator, perhaps sildenafil or inhaled nitric oxide, to see if that outflows the right ventricle and improves our success of support. I really want to thank the members of the ECMO lab. We have a tremendous team. I'm grateful to be a part of it. Also been fortunate to have fantastic mentorship, especially from Dr. Mialishka, who also happens to make a good rounding buddy on Christmas a few years ago. Thank the Department of Surgery at the University of Michigan for its support of this research, and of course, once again, the AMSA Foundation for the opportunity to present today. Thank you all, and I'll take any questions.
The education grant winner, if you attended the sessions yesterday, is Dr. Muiz Mustafa for development of a novel tool to improve surgeon readiness for rare diseases, the Just-in-Time Educational Collective. If Dr. Mustafa is here, please rise and be recognized. And then we have 2 global research scholarship winners. The first doctor I know is here, a roadmap for non-operative management of intussusception in Ethiopia, early steps toward widespread adoption. And Dr. Molla is an assistant professor of pediatric surgery and vice president of the Association of Ethiopian Pediatric Surgeons at St. Peter Hospital in Ethiopia. Dr. Molla, would you stand and be recognized? Thank you. And our second global research scholarship winner is Dr. Habib Ogundipe from the University College Hospital in Nigeria. He's a senior registrar in pediatric surgery, and his project is Euroflowmetric assessment of Nigerian children. He is not present here today, but again, I'd like to actually thank the foundation and our donors for being able to give 2 global scholarships this year. Thank you.